everyone. Welcome to the session number 36. My name is Sun Kang, and I'm the chair of the session. I apologize for my camera not working, and due to some personal circumstances, I was not able to get my camera working. I apologize again. First, I would like to thank the organizing committee for the opportunity to present our work. The title of the session is Integration of Wearable Data to Study the Interrelationships between Biological Processes and Mood in Real Time. Our session consists of the 3 talks. First, to showcase the processing of accelerometry data with GGIR in mMARCH, a Motor Activity Research Consortium for Health, then followed by compliance with EMA, and lastly, integrative modeling of accelerometry-derived features with major depression. Please leave your questions in the Q&A box, and I will follow up the questions after each talk. Our first speaker is Wei Guo. She is a biostatistician at the National Institute of Mental Health. She received her PhD in biostatistics from University of Hong Kong. She has established a pipeline to process high-level multidimensional mobile and wearable data to harmonize in multiple centers across different continents. Please join me in welcoming Dr. Guo. Wei, take it away.
Okay, let me share. Could you see my screen?
Yes.
Okay. Good morning, everyone. Thank you for the wonderful opportunity to present at the International Day of the Women in Statistics and Data Science community. In this presentation, I will be introducing mMARCH.ac, our package designed to streamline data processing after utilizing GGIR for accelerometer data. This package offers tools to simplify and enhance the management of accelerometer outputs, making the analysis process more efficient for researchers. Notably, mMARCH.ac has already been published in the Journal for the Measurements of Physical Behavior, showing its relevance and utility in the field. This is a growing body of evidence that underscores the importance of motor activity and sleep dysregulation as critical components of various mental disorders. In this context, accelerometers has become indispensable tools for exactly measuring physical activity, sleep, and circadian rhythms in real time. The Mobile Motor Activity Research Consortium for Health, mMARCH, is a collaborative network utilizing clinical and community studies. These studies employ standardized digital mobile protocols while collecting a shared set of clinical and biological measures, fostering a unified approach across participating research efforts. Therefore, we developed this mMARCH.ac pipeline as a streamlined, harmonized, and reproducible pipeline for accelerometer data analysis that could apply to all mobile data in mMARCH groups. As saw in this timeline, wearables have evolved significantly over the years, progressing from simple wristbands or bulky watches to more sophisticated and refined devices that offer enhanced functionality and design. mMARCH includes more than 10 centers worldwide, spanning countries such as Canada, China, Switzerland, Australia, and United States. Leading this effort is Dr. Mary Congas, who is based at this IMH campus in Maryland. In this project, we have collected accelerometer data using wearable devices like ActiveWatch and GeneActive. As a group leader, we provide guidelines and directly process accelerometer data for our collaborators. This necessity to streamline and simplify the workflow is what drove the development of this mMARCH.ac pipeline. And as a broader level, a key scientific goal across mMARCH studies is to better understand the interrelationships between physical activity, sleep, and circadian rhythms, and their impact on both mental and physical health in children, adolescents, and adults. Now I will walk you through the details of this pipeline. First, what's GGIR? The GGIR is a package developed by is widely used for processing multi-day raw accelerometer data collected by physical activity and sleep researchers. To date, GGIR has been successfully applied in over 90 published studies to handle accelerometry data. Building on this, we developed mMARCH.ac, our package specifically designed to post-processing accelerometer data after running GGIR. And this tool makes the workflow more efficient and user-friendly, particularly for researchers working extensively with accelerometer data. To begin, the input data of mMARCH.ac is the output generated from GGIR. So this left panel of the interface shows the folder containing the original bin files. GGIR processes these files and generates two main folders, meta and results. The meta folder contains CSV files with activity data, such as Euclidean normal -1, MO, and NGG. And the results folder includes summary files generated by GGIR, categorized as part 2, part 4, and part 5. Part 2 provides summary statistics for activity data, both at the subject and day levels. And part 4 contains summary statistics related to sleep. And part 5 consolidates various outputs into a comprehensive summary. So how does mMARCH.ac work? This slide illustrates the workflow. Following the GGIR approach, the mMARCH.ac groups its output into 7 modules, module 1 through 7, grouping functionalities in a logical processing order. Modules 1 to 4 are dedicated to data processing. Modules 5 to 7 are dedicated to producing R Markdown reports of data cleaning, feature extraction, and gel decomposition. Let's go one by one. And module 1 is a data preparation. The functions in module 1 focus on reading, merging, and transforming the raw accelerometer data for all subjects. The raw data is summarized using MO, a key measure of the volume acceleration. Once the data is processed, all CSV files are merged to facilitate the investigation of population-level data properties, enabling researchers to explore broader trends and insights across subjects. Module 2 is data summary. These functions of module 2 create a descriptive variables of day-level acceleration data for the purpose of data cleaning. And then module 3 is data cleaning, and these functions are dedicated to data cleaning. For example, days are more than 16 valid hours are flagged as valid days, and the subjects with more than 7 valid days are marked as valid samples by default. And module 4 is data imputation. In module 4, the missing MO data are imputed by taking the subject level mean over all the valid days for each subject at that time. And module 5 is data processing presentation. This function generates a comprehensive report in HTML formats. This report includes data quality checks, assessment of the completeness of GGIR output files, and analysis of missing data patterns, among other relevant information. And module 6 is check the non-wear time matrix at minute level. In module 7, the features extraction and visualization. In this module, 88 features are extracted from minute level activity data to characterize the domains of the physical activity, sleep, and circadian rhythmicity. These features are derived from outputs generated by GGIR and calculate using other R packages. Additionally, the standard deviation across days for each subject is computed for each feature. Participants' average are then calculated, stratified by weekday and weekends, as well as across all valid days. And I will go through this presentation in next slide. So just an example, using the clean data, we generate activity matrix and plot it in the data report. And this is from Module 5. This analysis included the following steps. Like the first one is the diurnal profiles of the raw data sets, and this were generated to provide a clear visual representation of activity patterns over a 24-hour cycle. And this one, a log transformation was applied to the raw data to stabilize the variance and manage skewness. And this one is the correlation analysis. We calculated the correlation to explore relationships between different activity measures. This step helps identify patterns and associations in the data. And the fourth one is the functional principal component analysis, FPCA. And FPCA is allow us to reduce dimensionality while capturing the most significant variation in activity patterns like PC1 to 4. And I lost some features attracted in Module 7. And for the first one is the time-related features, such as time duration for the sedentary, light, moderate, and vigorous activity. And the second one is the fragmentation. Fragmentation metrics examine the pattern of accumulation of sedentary and active time. And these metrics quantify how frequently and how long individuals alternate between sedentary and active modes. And the third one is the cosine linear model, a parametric approach used to study circadian rhythmicity by assuming a cosine-shaped pattern. And next one is the stability of rest activity rhythms, such as intra-daily variability and relative amplitude. This matrix assesses the stability of rest activity rhythms over time, capturing the degree of regularity in the individual's daily activity rest patterns. And the last one is the functional principal components. FPCA is used to represent the diurnal patterns through functional principal components, and this enables us to capture the primary modes of variation in the data, summarizing the most important features in activity patterns. As an output of Module 7, this concludes the summary of all features extracted across 3 domains, physical activities, sleep, and circadian rhythmicity. While these metrics provide a comprehensive overview of the data, I will not delve into details specific for each feature here, but I will show some examples. Here, this is our example study. Illustrates one of our heritability analysis using the output from mMARCH.ac pipeline. And the current sample comprises a subset of participants from the IMH family study of affective spectral disorders, recruited through the community screening in the greater Washington, D.C. area. In this study, we calculate the polygenic heritability estimates, which are defined as proportion of total phenotypic variance explained by additive genetic effects. These estimates were adjusted for age and gender using the variance components approach implemented in the Solar software. Notably, most of the active graphic features, such as sleep midpoint and the total active counts, demonstrated statistical significant heritability. Therefore, we can consider them as potential markers for the future association or the genetic association studies. And this is another example from our pipeline. And Dr. Kong will cover this topic in much greater detail. Essentially, once we have extracted all the cartographic features, it's too many, we can utilize Drive, the joint and individual variance explained software to decompose heterogeneous or accelerometer features into joint variation, individual variation, and residual noise. And these latent variables, referred as GEL scores, can be estimated and subsequently used as a novel measures in statistical analysis. And at last, how to use this software. To use mMARCH.ac pipeline, the first step for users is to install and load mMARCH.ac package. And here, and you can either load them from our Chrome or the GitHub. And next, users should run the create cell function, which generates a simple which is a very simple R script, and you can directly new all the files you needed. So for example, you can have this main call function, and you can customize by the users. Once customized, users can execute each module by running the corresponding R code within this script. And the parameters here, you can change the parameters as needed. Finally, as a summary, the mMARCH.ac package contains one primary function which does the following. Generates all necessary R markdown, cell executable files for the further data processing, and secondly, loads, reads, transforms, and merges the long activity data from CSV folder from GGIR outputs, and then creates descriptive variables of day-level activity data for the purpose of data cleaning, and then cleans the merged acceleration data and imputes the missingness by taking the average across valid days for each subject, and build a comprehensive report for data processing and explanatory reports generated by editable R Markdown files, and extracts multiple GGIR and non-GGIR features from the 3 domains. And then we can also perform an unsupervised decomposition of features of the 3 domains while GEL methods. And that's it. Thanks.
Oh, thank you. Thank you so much for the nice presentation, Wei. Do we have any questions from the audience? I don't see any questions in the chat box or Q&A box, so maybe we can move on to the next talk. Our next speaker is Ananya Swaminathan, and she is a postbac fellow at the National Institute of Mental Health. She received her bachelor's and master's degree in biomedical engineering from Johns Hopkins, and she hopes to pursue a PhD in biostatistics and bioinformatics. Ananya, the floor is yours.
Thank you for the introduction. Just my screen up.
Yes.
Sorry, do you see a window in the center of my screen?
Yes, your slides are fine.
Okay. Thank you to the committee for allowing me this opportunity to present. So today I will be discussing compliance with simultaneous collection of ecological momentary assessments and biological measures. Ecological Momentary Assessment, or EMA, is an ambulatory data collection technique that is designed to capture real-time data. It involves repeated self-report surveys and is usually administered in a naturalistic setting on a small portable device. EMA is typically used to capture data on mood and affective states. However, it has also been used to capture data on stress, physical activity, diet, and alcohol and drug use. Several studies have collected biological measures, such as salivary cortisol, in conjunction with EMA. There are many factors that can influence compliance with these procedures, such as compensation, stress levels of participants, and number of measures collected. However, most studies involving simultaneous collection of EMA and salivary cortisol involved collection periods under a week, and many of these studies restricted their samples to adults without psychiatric disorders. Therefore, our goal was to examine the feasibility of collecting simultaneous salivary cortisol and EMA measurements in a naturalistic setting over a longer period of time, in our case 2 weeks, in a sample that has been enriched for mood and anxiety disorders, and in a sample that includes participants at different stages of the lifespan. So participants included here were part of the NIMH Family Study of Affective Spectrum Disorders, which is a community-based sample that has been enriched for mood and anxiety disorders. A subsample of this group, including 153 participants, had valid cortisol and EMA measurements. The average age of these participants was 37.4 years, with a range from 11 years old to 84 years old. 59.5% of the sample was female, and of the 153 participants, 20 had bipolar 1 disorder, 17 had bipolar 2 disorder, 46 had major depressive disorder, and 35 had only anxiety disorders, so no mood disorders. Additionally, 13 participants had other disorders, so these included eating disorders and substance use, but they did not have mood or anxiety disorders. Lastly, 22 participants were healthy controls. These participants completed EMA and salivary cortisol measures 4 times a day for 14 days. Cortisol values below 0.037 and above 1.85 micrograms per deciliter were treated as invalid or missing, since these values were considered biologically implausible. Missingness for both EMA and cortisol was assessed stratifying by age group, sex, and mood and anxiety disorder group. In our results, we found that age group significantly impacted compliance, so we decided to explore this further by creating generalized linear mixed models with a random intercept for each subject to determine how interactions between age group and time of day of week, and day of collection period impacted missingness. We calculated probabilities of compliance by holding other variables at fixed values. There were a total possible of 8,568 EMA and salivary cortisol measurements for the 153 participants. Of these total possible measurements, 6,638 EMA measurements and 5,646 salivary cortisol measurements were completed and considered valid. Using chi-squared test and pairwise proportion test, we found that the youngest age group, the 10 to 20 age group, was significantly less compliant than the other 3 age groups for both cortisol and EMA measures. We also found that females were more compliant than males, and for the mood and anxiety disorder groups, we found that healthy controls were significantly more compliant than participants with bipolar 2 disorder, major depressive disorder, or anxiety for both cortisol and EMA measures. We decided to look further into how these interactions between age group, time of day of week, and day of collection impact missingness. So we started by looking at EMA compliance by age group and time of day. Doing so revealed that the 10 to 20 age group was the least compliant across all 4 time periods across the day. This group was particularly non-compliant during the midday assessment when probability of compliance dropped to almost 50%. We then looked at EMA compliance by age group and day of week. Again, we found that the 10 to 20 age group was the least compliant. However, there were no day of week effects for any of these 4 age groups. We then looked at EMA compliance by age group and day of collection. We found that for the 20 to 40, 40 to 65, and 65 to 85 age groups, compliance either remained the same or increased across the collection period. Meanwhile, for the 10 to 20 age group, compliance decreased across the collection period. We also looked at compliance by age group and mood disorder. Again, we found that the 10 to 20 group was the least compliant, but we also found that the 20 to 40 age group was the most compliant. And we found that controls were slightly more compliant compared to people with any disorder. Lastly, we looked at EMA compliance by age group and season. This revealed that people were slightly more compliant during the autumn than they were during the other seasons. We also looked at these factors for cortisol. So when examining cortisol compliance by age group and time of day, we again found that the 10 to 20 age group was the least compliant. However, we also found that for the 20 to 40 and 40 to 65 age groups, that their compliance decreased for the evening measurements. When looking at day of week, we found that the 20 to 40 age group was the most compliant across all days. And we found that generally the 10 to 20 age group was the least compliant. However, unlike with EMA, we found a weekend-weekday effect in the 10 to 20 age group for cortisol measurements. So as you can see here, compliance is quite low during the weekdays, but increases for this age group on Saturday and Sunday. We also looked at the effects of age group and day of collection. Once again, we found for the 20 to 40, 40 to 65, and 65 to 85 age groups that compliance either remained the same or slightly increased across the collection period. And for the 10 to 20 age group, compliance decreased across the collection period. As with EMA, we found that controls were slightly more compliant than people with any mood disorder. And we found again that the 10 to 20 age group was the least compliant. Likewise, we found that for cortisol, people were also more compliant during autumn compared to the other seasons. So these results demonstrate the feasibility of simultaneous collection of ecological momentary assessment and salivary cortisol in a sample that includes participants across the lifespan and is enriched for mood and anxiety disorders. There are certain procedures that could be implemented to improve compliance. These include increasing the time window for responding. So participants in this study had 20 to 30 minutes to respond to EMA prompts. So increasing the length of this window could potentially increase compliance in case participants aren't available during the first 20 to 30 minutes that they get an alert to complete an assessment. Another way to increase compliance could be implementing incremental compensation. So this would involve, rather than just compensating participants based on whether they completed the EMA or not, there could be a certain base level compensation that increases based on the number of assessments that people have completed. Another method that could improve compliance is flexible scheduling. So this could allow participants to customize their schedules to times that work best for them, and hopefully that would improve compliance since they would be tailoring it to fit their own schedules. Lastly, we were thinking of having special accommodations for youth. As I mentioned before, for the cortisol measurements, there was a bit of a day of week effect in the younger group. So the 10 to 20 age group, they were less compliant on weekdays compared to weekends. So one way to account for or to kind of counteract this is to coordinate with schools to make sure that youth can access their assessments and collection materials during school hours. Thank you.
Thank you for the nice presentation, Anna. Yeah, it was interesting to find there are like different missingness and compliance rate across the different age groups and different timing of the assessments. So I was wondering if there's any questions from the audience. Do we have any questions from the audience? I don't see anything on the chat box or Q&A box. So then I will move on to the next talk, which is mine. And let me share my screen. Can you see my screen?
Yes.
Thank you. My name is Sun Kang, and I'm also a statistician at the National Institute of Mental Health. And I'd like to talk about the joint and individual variations of sleep, physical activity, and circadian rhythmicity features in COLA study. As we already showed these figures, the aim of the study is to examine the joint and individual variations of the actigraphy features and their associations with mood disorders in a large population-based cohort study in Lausanne, Switzerland. And I'd like to mention that COLAS is a part of mMARCH, a Motor Activity Research Consortium for Health. And there are 6 core sites involved in mMARCH. And the first one is the COLAS Psycholas study in Lausanne, Switzerland. And there is a Netherlands study of depression and anxiety at VU University Medical Center in Amsterdam. And the third one is the Youth Mental Health Clinic at the Brain and Mind Research Institute in Sydney, Australia. And the fourth one is the Brisbane Adolescent. And then the Hong Kong high-risk study of bipolar I disorder at the Chinese University of Hong Kong. And lastly, the NIMH family study of affective and anxiety spectrum disorders in Bethesda, Maryland. The core group selected the gene-active device to measure the actigraphy, and most sites also include the EMA, the Ecological Momentary Assessment. The mMARCH network reflects the growing interest in assessment of these rhythms in daily life. And as Dr. Guo has presented in the first talk, we have developed standard procedures for study administration, data management, and analysis of actigraphy data, and also established procedures for the administration using the mobile applications, processing and analysis of the EMA. We also conducted a cross-site and meta-analysis in order to increase power and cross-validate common, and also established a work group to develop database with environmental light, temperature, and other exposures to prepare for analysis of the influence of the climate changes on mental health and behavior. This collaborative work has grown. Like, for example, EMA has been available in English and French, but now we have translated it in Korean this year and working on to extend the application in other languages as well. And the background of this study, there is a now growing evidence of dysregulations of sleep, physical activity, and circadian rhythms in people with major depression. However, few studies of large population cohorts have examined associations between the full range of the features that can be derived from actigraphy. And here we examine the interaction and intercorrelations among the 3 major domains extracted from the actigraphy using GGIR method to quantify their joint and individual variations. Here are the main questions of today's talk. What is the overlap between the domains of the sleep, physical activity, and circadian rhythmicity assessed by actigraphy? And whether, are they correlates of mood disorders, and are they state or trait markers? The study sample included 2,306 participants from a prospective cohort study selected from the general population. The mean age of participants at the time of the latest interviews about 62, with ranges from 45 to 86, and about 54% of the participants are female. Clinical diagnoses were obtained through a comprehensive diagnostic interview to ascertain lifetime history of mood and disorders. There are a total of 1,145 people with major depression, including 184 with chronic depression and 961 with remitted major depression and 1,161 with no history of major depression. Features of sleep, physical activity, and circadian rhythmicity were accessed from using actigraphy collected for an average of 12 days by a wrist-worn accelerometer called Active. And these are the features of the sleep, physical activity, and circadian rhythms domain included in the study. So in sleep domain, we included timing of the sleep, which is sleep onset, wake up, duration, and midpoint. Midpoint is the middle point of the onset and wake up. And then we included sleep duration. And then we included features in quality, which is sleep efficiency and number of sleep blocks during the nighttime and number of the wake up at least 5 minutes during the sleep period. And then in the physical activity domains, we included total volume of the physical activity, which is represented by total activity count and total log transformed activity count. And then also total sedentary time and duration of the light active physical activity, as well as duration of the moderate to vigorous physical activity, and also fragmentations of the physical activity, which includes sedentary to active transition probability and active to sedentary transition probability. And we also included several circadian rhythmicity features, which are represented by the strength, stability, timing, and diurnal temporal landmarks. Relative amplitude, M10, L5, and measure amplitude. And those are measure of the strength of the circadian rhythms. And stability of the rhythms are represented by inter-daily stability and then intra-daily variabilities. And then the timing is, M10 timing is the timing of the middle of the timing of the most active 10 hours and least active 5 hours, etc. So GGIR is the abbreviation of joint and individual variation explained, and this method was applied to derive the joint and individual variance of the derived features in 3 domains. And GGIR actually decomposes the original multi-domain data into a sum of the 3 components: rank approximation, capturing the joint variation of the domain, and then the low rank approximations, capturing the variation individual to each domain, and then the third, residual noise. The imposed rank and orthogonality constraints can be considered as extension of the PCA, the principal component analysis. And then the full details of the GGIR and its estimation can be found in the references below. And GGIR provides a non-parametric low-rank-based procedures to estimate and exclude joint across domain variations from the original features by creating a adjusted domain-specific features. For example, it is possible to filter out the effects of the physical activity from the quality of sleep, which regular PCA is not capable of. And row orthogonality was enforced both between joint and individual components. So GGIR is fitted by Interrelationships actively estimating joint and individual structures. And before we apply GIRE, we created within subject level averages and within subject level variability features across the 3 domains, and then normalize those scores to use as input for GIREs. And then these steps was also implemented in mMARCH.ac that Dr. Wade will present in the first talk. So optimal choices of the low rank representation were estimated to be rank 4 joint structures and rank 1 structure individual to the physical activity domain, and rank 1 structure individual to sleep domain, and rank 3 structures individual to circadian rhythmicity domains for average features. Whereas for the subject variability features, there are 2 joint components and 1 individual sleep component, 2 individual physical activity component, and 3 individual circadian rhythmicity components. The figures in this slide displays a bar chart of the amount of the variation explained by joint and individual components in each of the 3 domains. And interestingly, more than 80% of the variation in physical activity and around 57 to 66% of the variations in sleep and circadian rhythmicity can be explained by the joint components and individual for the average features. And individual components explain only around 29, 11, and 28 of the total variation in physical activity, sleep, and circadian rhythmicity domains, respectively, for the average features. And on the other hand, only 47, 55, 36% of the variations in sleep, physical activity, and circadian rhythm can be explained by the joint components with the variability features, which are much less compared to the average features. Unlike the original features that demonstrate significant correlations, the derived GIRE scores are orthogonal by construction. And like how we do PCA scores used in the principal component regression to substitute the original covariates within PC scores, GIRE scores can also be used in the same way in regression modelings. So this slide shows the GIRE regression results. Because all scores are uncorrelated and normalized, the magnitude of the regression coefficient can be approximately interpreted as a contribution to the increase of the adjusted R-square. And what are the interpretations for the significant component in these models? We need to look at the loadings of each component. So, for example, joint 1 and jive joint 1 and jive joint 2 are significantly associated with the individuals with depression. But what does it mean? And joint components and joint information was dominant across the 3 domains of the sleep, physical activity, and circadian rhythmicity, and which highlighted in the importance of the joint consideration of these domains. And what is interesting in that the first joint component, the most important component, measures time spent in high-intensity physical activity and strength of the circadian rhythms, and especially amplitude, that are highly intercorrelated. And this means that it is unlikely to be same time have a weak circadian rhythm measures by the rhythm amplitude. And the second joint component capturing timing of the 24-hour landmarks, especially timing of the sleep and timing of the peak of the physical activity. And so these 2 joint components are associated with both current and remitted individuals with MDD. And so this is the a summary of the jive regression results showing that joint 1 and joint 2 associated with the both MDD current and remitted individuals, whereas joint 4 is only associated with the MDD current individuals. And then below shows the top 5 features with the highest loadings of each component. And here, only jive joint 2 component is associated with the individuals with MDD and depression when we apply this method for the variability features. And here are the results for the jive regression with the individual features, showing that circadian rhythmicity, first for the average, first for the average features, individual circadian rhythmicity component 1 and 2 are associated with the MDD lifetime, but the first individual component in circadian rhythms associated with remitted MDD, whereas the second individual circadian rhythm component is associated with the current MDD. As for the variability features, circadian, again, the individual one component is associated with the remitted MDD, whereas the second individual circadian rhythmicity component is associated with the current MDD. So here are the findings. Joint variance strongly outweighed individual variance of the domains extracted from the accelerometry of the average features. And then the first and second joint factors distinguished characterized depressive traits, whereas the fourth joint component indexed depressive state. For variability features, individual variance outweighed joint variance of the domain compared to the average features. And then the first circadian individual factor characterized depressive traits, whereas the first sleep and second circadian rhythmicity individual factor indexed depressive states. And there are some limitations. These findings are based on only 2 weeks epochs of the actigraphy that may not reflect stable patterns of these variables. And findings may not generalize to different age, ethnic, or geographic subgroups. Despite the consistency in findings from the accelerometry in mood disorder across the adults, they may not extend to children or adolescents. And also, we could not evaluate the potential effects of the medications on patterns of the activity identified in these analyses. Even though many prior studies of the accelerometry in mood disorder have not adjusted for the medications, our post-hoc analysis that adjusted for medication use in fact did yield some differences in the findings, and confoundings between the medication use and severity complicates the interpretation of these results. So future studies that systematically test the effect of the medication on objective measures of the physical and activity and sleep may be needed. And so these are the conclusions. And the most important messages we'd like to propose is a need to think about the 24-hour human behaviors, including sleep, physical activity, and circadian rhythmicity as a whole, and try to better understand the interdependencies between these behaviors, both from a system biology and clinical perspectives. And currently, these 3 areas, the physical activity, sleep, and circadian rhythmicity are divided into different disciplines to some extent. And our work shows the need for more global 24-hour perspectives. And I'd like to thank our colleagues and collaborators. And thank you for listening to our thoughts. Do we have any questions? Okay, I need to promote the next session call first. Okay. Okay. Thank you everyone for joining our session, and I will end the session now. Thank you.